Oh yeah, this is going to be recorded, so if you have your lab, then don't you're not going to miss anything. But hello, once again, final review, week 15. It's crazy. Oh yeah. So firstly, do we have any preliminary questions? I remember the genetics lab final being so much easier than any of the labs that we did, but that was back when I was online. Cool. Okay. Oh, let me turn my camera off. Ew. No, I said turn off, please. There we go. Okay, so question time. Where are we? Oh yeah. Sorry. Okay. Question number one. These are all pertaining to what we've done in the last week, for the most part only. So, excuse me. During translation, what kinds of molecules are being chained together? And what kind of chemical reaction characterizes these chains? Yes, Usman, these are amino acids that were being chained together. Let me even find um, so it's kind of like we're talking about all these processes with words and pictures, and that's just like a really terrible way to do it because this is a moving process. So this is what God loves it. Okay, let me find a better one. Sorry. This is what's happening in translation. We have this huge purple blob is the large and small ribosomal subunits. And then we have on the bottom here, this is our mRNA strand from 3' over here to 5'. Let me try to make this bigger. Okay. Okay, so we have the mRNA strand from 3' to 5', and then we have here, we see this little like cylinder looking thing, and we see that it can fit within it 3 nucleotides. These 3 nucleotides are our codons. And this thing is kind of like what we call the frame, especially when we're talking about frame shifts. And this is in the cytoplasm, right? Yes, because we remember from the central dogma of biology that we have DNA that's transcribed into protein or into RNA. And this RNA exists for the most part as our mRNA or our rRNA, which is the ribosomes that you find in the rough endoplasmic reticulum and floating around in the cytoplasm. So yes. And so we have some rRNA, our ribosomes, and then we have our mRNA, which is this strand down here. And then what we have are these these cruciform things. And this is our tRNA that holds our 3 anticodon, or not our 3 anticodons, our anticodon CUA, which relates to this GAU. And so this is what's actually happening. We have an anticodon that's attached to a tRNA that's attached to an amino acid. And what happens is when you go from the E site to the P site, the amino acid gets linked into this chain. We see that tRNA comes on, and then it attaches this amino acid to the growing amino acid chain. And yeah, it's just kind of hard to visualize with static pictures and stuff. Yes, like Usman said, we are chaining together amino acids. And what kind of chemical reaction characterizes this blocking together of amino acids? This is a recall to Chemistry 213. No problem. Can you guys like hear my background? There's kind of some noisy stuff going on. Okay, good. It's no problem. This is, it's a long time in the past, kind of maybe. The answer is dehydration, because when we have our, so firstly, what is this process called? Bro, I knew, trust yourself, Usman, trust yourself. So we have these amino acid monomers. I wish I hadn't gotten rid of it. And when you chain together a bunch of individual monomer units, what is that resulting chain called? Not exactly, Casey. Yes, thank you, Carla. It is a polymer. So when you have multiple monomer units put together, you form a polymer. That's what a polymer is. It's just a chain of individual monomer units. And these monomer units, so we have our amino acids. What happens is different amino acid, or I guess all amino acids, have an OH and a H, like on the side. Right, so if we have our amino acid monomer units, they're kind of like stuck within a hydrogen and an hydroxide. And so what we need to do to link these two monomers together is get rid of the H and the OH on the inside. And so when we do that, we end up with, so we pretty much cut this off, and then we cut this off. So what we end up with is the HAA tied to the AA tied to the OH, because we just took off the ones that were separating them, not the ones at the edge. And what happens is this OH and this H comes together to become H2O. That's a 2O. And so what we did is we kind of like took a water molecule out of this system. And in order to make the polymer, we took a water out. So we dehydrated it. Yeah. Yep, dehydration. That's just some recall to 213, but we talked about it and I wanted to mention it. Oops.
All right, so now we can go ahead and talk about what the structure of an amino acid is. So does anybody want to tell me what an amino acid looks like in terms of like its chemical structure? Yes, it has an R group. What is the R group attached to? All right, it's attached to a central carbon. The carbon is like the center, and then you have an R group. And what other, someone said carboxyl, which is one of the other things attached to that carbon. What are the two other things that we'll find on our central carbon of an amino acid? Yep, a hydrogen. And we're missing one more thing. Not necessarily a methyl. A methyl can be found on an amino acid, but that's a type of R group. All right, I'll just give it to you. It's an amino group. So on your amino acid, you have an amino is like kind of like a, oh my gosh, what am I trying to say? I don't know what I'm trying to say. But anyways, so you have your 4 groups. And so you have your amino group, which is like why it's called an amino acid. And then you have your carboxy terminus. So that's your COOH. And then you have a hydrogen. And then you have your R group. So I think it was Carla who asked if it was methyl. Methyl is a type of R group that you can have on your amino acid. And so basically, these R groups give your amino acid like its very specific characteristics. And so we saw that chart in class that had the 5 categories of different amino acids. And so you can have non-polar amino acids, aromatic amino acids, polar uncharged amino acids, and then positive and negative amino acids. Carla asked, do we have to know the ones that have hydroxyl groups? I cannot recall if Dr. Chiari said whether or not she wants you to know that. Garrett, do you remember?
The only things that were in the slides were just recognizing or being able to replicate the like central carbon attached to a hydrogen attached to an amine and carboxyl group with an R group on the bottom. I think that's all we had.
Okay, I thought so. So no, you don't have to know which ones have hydroxyl groups. You don't have to know like the structures and stuff of specific amino acids, but you need to know that in general, you have your amino group, carboxy group, hydrogen, and your R group. And so something that's important about the amino group is that it indicates the beginning of your protein. And so the end of your protein that has the amino, like a free amino group that's not attached to anything, that's called your N terminus, and that's the start of the protein. And then all the way on the other end of your protein, you'll have a free carboxy terminus, which means it's not attached to anything. And that indicates that it is the end of your protein, because neither of those, the amino group and the carboxy group, are not in a bond, a peptide bond. So pretty straightforward, but does anyone have any questions about what an amino acid should look like? All right. If not, Garrett, you can take it away.
Sick. Okay. So this would be so much easier if I could draw it, but how does the first degree structure of protein relate to future degrees of structure, like second and third and fourth? The formation of hydrogen bonds. Yes, that is the key component. That's actually something I failed to mention, but largely, like degrees of structure, it can seem kind of confusing because like first degree, second degree, who's putting math inside my biology? I don't like it. I'm uncomfortable. Oh no. But really, it's just like increasing degrees of complexity. So first degree structure is, an arrangement of monomer units into a polymer. First degree structure is just like the sequence that you have. Second degree structure, is when you put 2 sequences together. For example, you have a polymer could be 1 strand of DNA. And then when you put 2 strands of DNA to form a double helix, that is a second degree of first degree does not have hydrogen bonds. First degree structure is held together by phosphodiester bonds. Well, that's in the case of DNA. It's different for different things. I'm just kind of talking about like general first, second, third degree structures of proteins. And hey, Joseph, what's up? Yeah, peptide bonds, specifically for DNA, or actually no, specifically for proteins, and phosphodiester bonds for DNA. But yeah, so second degree structure is just like putting 2 chains together. And third degree structure is when you have this like double helical structure, and then you kind of like make different shapes with it. I'm not explaining this well, but like Usman said, when you have hydrogen bonding from one double helix to another, then they can like combine and make super funky shapes. Let me try and get a picture to exactly. So we have just the sequences, right? We have our, these are actually amino acids right here, because we have our R group, our H group, and our amine group attached to a central carbon. And so we have just the chain of stuff, you know, this is our polymer. And then when we put a bunch of polymers together and they hydrogen bond with each other, they can create some funky shapes. This is our secondary structure because you just have, you know, these individual polymers, these individual first degree protein structures that connect to each other. So an increasing degree of complexity is that you have just the chain and then a group of chains. And then graduating from secondary to tertiary structure, you go from a group of chains to a group of groups. And this is just a bunch of double helixes put together. And then quaternary structure is we see this orange unit. We just have a bunch of orange units put together. So increasing degrees of protein structure are just increasing degrees of complexity of the same kinds of, you know, things. I kind of mentioned it a little bit, better diction here.
I'm sorry, I have a quick question.
What's up?
So just to clarify, the primary structure is just like the amino acids by themselves, and then the secondary structure is them being combined through hydrogen bonds?
Exactly. Very well said.
Okay, thank you.
So the primary structure of DNA is, for example, this. Okay, so if we have no other questions, oh, I should not highlight it. If we have no other questions, let's move on to number 4. What two qualities of the genome give it its power? And when I say power, I'm asking, like, what about DNA has made it such an advantageous and flexible molecule such that the most dominant organisms on Earth throughout Earth's biological history have used DNA as their genetic material? This is also a recall to Bio 213, but we did mention it in class on Tuesday. Evolution, that's a very good guess. That is how DNA acquires changes and helps organisms to, there we go, Joseph. Well, that's one of them. So Joseph mentioned redundancy, the redundant nature of DNA, and that is very correct. What is the redundant nature of DNA? You named it, but what does it do? It's like where many codons code for a single amino acid. Excellent, excellent, excellent. So DNA's two most important qualities are that it is redundant and that it is unambiguous. Unambiguity, so ambiguous is like, you know, having multiple different things that can happen, because ambi means multiple or lateral, I think. Never mind, let's not get into the etymology, but you know, if something's ambiguous, it's not certain. If it's unambiguous, it is certain. So DNA is unambiguous in the sense that if you have UGA, it is going to code for stop. It's not going to code for methionine. Yes, thank you. Both is what, thank you, Joseph. Ambi means both, apparently. So for example, if we have UGA, that's going to only code for stop. It's not going to code for leucine. It's not going to code for glutamine. It's not going to code for methionine. It only codes for stop. However, so that's the fact that one codon only codes for one protein. However, it is also redundant in the fact that a bunch of different, that a particular protein can be coded for by several different codons. So the codons that code for this protein only code for that protein, but there are multiple things that code for the protein. Let me show you. So if we have the codon CAU, it will unambiguously code for, actually I don't know what this amino acid is. So let's try another example. If we have GUU, this codon will only code for valine. It will not code for alanine or asparagine. Oh, histidine. Thank you, Carla. So GUU will only code for valine. It will not code for any others, not serine, not proline, none. However, multiple codons can make the protein valine. This is redundancy. So if you have, for example, if the original sequence was GUU and you have a mutation at this third nucleotide, no matter what it mutates into, whether it's cytosine, adenine, or guanine, it will still produce valine. It will have that redundant failsafe to still produce the same protein that we need, the same amino acid. And that flexibility is why DNA is so advantageous. Histidine, I need to remember that.
Oh my gosh, unfortunately, all of the amino acids have been hammered into my brain. Yeah, me too, Carla. All right, so kind of switching a little bit. So we talked about the Shine-Dalgarno sequence during class. And so can anybody tell me what exactly it is and like why we even care about it? Yes. So yeah, Joseph got it. So it's a recognition site. It is a place where the ribosome can recognize like that very specific sequence and it knows where to bind. So your ribosome binds to the Shine-Dalgarno sequence in addition to the start codon. And so the reason why we can't call it a promoter factor is because it's not like a different structure that comes in and helps the ribosome bind. Like it's an inherent part of, or an intrinsic part of the actual mRNA itself. And so can anybody tell me where this sequence is? All right. Yes, so it's in the mRNA. And can anybody tell me specifically like where in the mRNA it's found? So like, is it after our start codon or is it before our start codon? Before. Yes, so it's before our start codon. So since it's before our start codon, it doesn't actually get translated into an amino acid. It's just like a place that helps us know where we are and where to bind. So what do you guys think would happen if we mutated the sequence? Would we still be able to bind the ribosome to the mRNA, or would we not bind? Would we maybe go in the wrong direction? So yes, there is a chance that transcription might not start, but because your ribosome binds to the Shine-Dalgarno sequence and the start codon, there's a chance that it could still bind to your start codon, but the Shine-Dalgarno sequence kind of gives you orientation. So it tells you, okay, so the ribosome should read from left to right instead of right to left. So if you don't have that sequence there and you only bind to your start codon, you might end up reading in the wrong direction and translating in the wrong direction. So translation might still happen, but it also might go in the wrong direction. So does everyone feel okay with that? Okay, cool. Garrett, you can take the floor then.
Okay, friends, we have an mRNA sequence. However, well, you can take the time to read it if you want, just to get familiar with it. And so what we have is that this original mRNA sequence accidentally has its first uracil deleted. So how do you, wait, get out of here. So the first uracil is accidentally deleted during splicing. What has just happened? And what is the effect? Very good. It's a frame shift mutation. So I guess there are two parts of the answer. What happened? The first like general thing that happened, what is that? And then the second thing that I'm asking is what products, like what change in the products occurred? It'll change the amino acid sequence. Exactly. So we have this sequence of RNA. And what's going to happen is that this RNA is fed through your ribosomal subunits, and it is going to be read in frames. So tRNA attaches via the anticodon to the codons of your mRNA. And the tRNA, kind of like what I showed you in the GIF, it looks at a specific frame, kind of like if it was only looking through a window frame at the mRNA that's inside. And so it can only, or the window frame is only wide enough to see 3 nucleotides at a time. And so what happens if we just delete this guy is that the frame has shifted. The window frame, when it used to only be able to see AUG, now it looks at the 3 nucleotides at a time. And now this is AGU. So we no longer have our start codon. And so everything is just going to change. If it can't find another AUG within the sequence, I'm not even sure if it could start. However, we do have an AUG, fortunately. So yes, the window frame that tRNA looks through to look at the mRNA, excuse me, it shifts. And so the removal of this nucleotide shifted the frame shift mutation, and it shows different codons to the tRNA. So now entirely different amino acids are translated. And so let's look at this codon table. Let's zoom out. So we have, or we had AUG, that's methionine, the start codon. And then we have UUU. Oh, and it's important to note that I'm not sure if you'll be given questions about this really, but like going from a DNA sequence to the mRNA sequence to the anticodon sequence, because you have to like switch back and forth because mRNA is complementary to the DNA, not even perfectly complementary. You have to switch all of your A's for, or your T's for U's, and then your anticodons are complementary to your mRNA sequence. I'm not sure if you'll get questions about that, but be mindful that codon tables contain the nucleotides that you see in the mRNA. So I kind of made it easy on you. So we started with AUG, that's our start codon, UUU, which is, I don't even know, and then PHE, whatever that is, GAU, GAU is asparagine, and then GGC is glycine, and then UGA is one of our stop codons. So our original sequence was this. Excuse me. So how has it changed now that we delete this? Well, now we have this AGU. Is this going to start translation? There's no start codon here, exactly. And then, so this is one frame. This, get out of here. This is another frame. Is there a start codon here? No. So again, this frame is just out of the picture. This frame is also out of the picture. Actually, I should make them different colors. Oh, but what's this? We have an AUG. So finally we can start. Thankfully, there is an AUG here. So we now have our methionine. Here's the first frame of our sequence. And then the second frame is GCU. So GCU is ALA. And then GAA, which is glutamine. And then another AUG. Oh no, another start codon. And then UC, and then the sequence ends. So there is no even, there isn't even a stop codon in this new sequence. So this deletion that happened, which caused the frame shift mutation, could have very significant consequences. Because if this sequence just kept going and there was not a stop to put it in its place, then it could just like keep going and going. And also, this is not what our cells originally intended to make, because we really needed this amino acid sequence to perform a specific function within our cells. But now we get this. So something's wrong, something's not good. So this is an example of a frame shift mutation doing something that's probably going to end up killing your cell because it doesn't have the amino acid sequence that it needs. Did that make sense to everyone? This should just honestly be practice. Yes, okay, good. Oh yeah, Sam, I think you're muted.
That's awkward. Sorry. Thank you for letting me know, Garrett. So basically, this next question is really similar to the last one. It's just being able to like, it's asking you guys, can you figure out what the amino acid sequences based off of a given mRNA sequence? So does anybody know what our first amino acid would be? I'll make this table bigger so we can see. So Usman says AGG, not quite. Does anybody else want to give a stab? Okay, yes. So Carla got it. This was kind of like a trick in a way. So we have these three codons before we get to AUG, which is our start codon. So you always have to start with AUG. And so these two that come before don't even matter because there was no start codon before it. And so we start with methionine, And then can anybody tell me what the next one would be? Yes, we'd have serine. And then after serine, we would have arginine. And then the last one, yes. So we'd have glutamine, which corresponds to this UGA. And then we have stop. So stop was GCG. And our stop codons don't actually code for an amino acid. It just tells your ribosome to stop translation and it releases. So we have 3 different stop codons. So stop can be indicated by UAA, UAG, or UGA. So, oh, my bad. I misspoke. So UGA was stop, and then GCG is just extra at the end that we don't really care about. But yeah, UGA is stop. And
So we just ignore the first 2 and the last.
Yeah. So you ignore the first 2 because you haven't started yet. And then once you get to your start codon, we care about all of that. And then once you get to your stop codon, everything after that, we also don't care about.
Yeah, because your cell is only going to or your RNA is only going to do what it's told to do. And so if it doesn't receive the signal from, or if it doesn't detect a methionine to tell it to start, nothing before it is going to be translated.
So does anybody have any questions about mRNA and translation and any of that stuff?
Do you think there will be a question where they give us one sequence and then we have to get the mRNA sequence from that and then get the amino acids from that? Or are they just going to give us their mRNA sequence straight up?
Well, I'm sorry.
Oh, sorry. I was just going to say, I don't think she'd give you the amino acid sequence like methionine, arginine, leucine, because there are many different codons that code for each of for leucine and whatever else I said. So I think that you'd probably see an mRNA sequence and be asked for the amino acid sequence.
Yes.
Oh, yes.
Sorry.
Sorry. Here. And Usman says he's confused about the initiation factors in eukaryotes and prokaryotes. Garrett, do you want to go over that or do you want me to?
Excuse me. Initiation factors in eukaryotes and yeah, you've got that. But firstly, if I could please, just to make sure that we're not struggling to perceive it, but this is kind of a more palatable way to think of or to visualize translation. So you have your tRNA, the orange guy, and then it connects via the anticodons to the codons that are part of the mRNA sequence. And then when they jump in, a peptide bond forms between the individual amino acids that are or that were part of, or excuse me, connected to the tRNA, and then it moves from the A site to the P site, and then it exits via the E site. And yeah, Oh, I can put it in the chat. Copy. Where's the chat? Okay, there's the chat. Yes, on to Usman's question. Did you want to talk about that, Sam?
Sure. Let me pull up the slides.
Which chapter is that?
I think it's sure. Well, actually, that's a good question. Usman, are you confused about transcription or translation? Okay, so the last one we did is translation. So that's chapter 15. Okay, I have the slides pulled up right now, Garrett. And okay. So let me see. Okay, so basically in prokaryotes, you have 3 initiation factors. So you have IF1 through 3, and these are separate. Are they proteins? I think they're proteins. Anyway, so they're separate proteins that come in and help your ribosome actually bind to your mRNA. So they're not part of your mRNA sequence and they're not part of your ribosome. They're just like their own things that kind of like float in and help everything stick together. And so for prokaryotes, those are the 3 initiation factors that you have to be concerned about. But then when you have eukaryotes, because of course it just has to be more complicated because why not, I guess. You have many, many more initiation factors. And so we don't have to know the details of like all of the individual ones, but you do need to know that you have 12 of them and we have more of them because eukaryotic translation is more complicated. Yes, slide 45. Was there something specific about slide 45 that you wanted to talk about? Okay, so just like go over slide 45 as a whole. Yes, I can do that. So your ribosome has 2 different units and those units stay separate. And when they come together, that's when translation actually happens. And so it would probably help if I zoomed in and made this bigger. So basically what happens is you have your mRNA with your Shine-Dalgarno sequence and your initiation codon, and that's how our ribosome knows where to bind, like we were talking about earlier. And the first thing that happens is your small subunit. So that's obviously the smaller subunit of your ribosome is going to bind to that sequence. And you have one of your initiation factors come in, which is IF3, and it helps your small subunit bind to your mRNA. And then after that, you get your first transfer RNA, so tRNA that comes in with methionine, which is your first amino acid. And it binds to your ribosome with the help of your other 2 initiation factors. So that's IF1 and IF2. And once that happens and like everything is attached, you also have a GTP that comes in that provides energy to actually make your bonds. Then you can bring in your large subunit, which is the bigger subunit of your ribosome. And that's pretty much like the layman's term gist of how this works. Okay, cool. I'm glad that it makes more sense now. Was there anything else about initiation factors and stuff that you wanted to talk about? Or are you all good there?
Could I share my screen really fast?
Mm-hmm.
So this is what's with that. It doesn't show the initiation factors, but this is the process Sam was just talking about. We have our mRNA sequence, and then our depending on this, it depends on the size. I think this is prokaryotic because it's 3050, but don't quote me on that. So we have our mRNA, and then our small ribosomal subunit attaches at the bottom, and then the tRNA comes in, and then the large ribosomal subunit sits on top over that. I was just eating a whole bunch of stuff with my, yeah, no problem. I was choking on an orange while you were talking about DNA. So no problem. Very relatable. Do we have any other questions?
Do you
Oh,
guys think it'd be possible to do like another example of like the genotypic frequencies and allelic frequencies? Because like I struggled with that on the exam.
oh yeah. Let me find it really fast. Oh, this should also be exam 3. Okay, so genotype this actually, could you please hold that thought and we will come back to it? Because there was actually someone who had some very pressing questions as well. And this is it would take, would you mind if we did someone else's questions first?
Yeah, that's okay.
Awesome, thank you. I'm sorry. Okay, can you see my screen? Or no, I'm not sharing. Okay, so we had some questions. Ugh. So these are questions from, I think, the second exam, question mark? No, this is the first exam, excuse me. And this was about some dominance stuff. And this was a question that really tripped everyone up for the most part. God, it's been such a long time. So a person contacts a breeder to buy a purebred dog for the following characteristics: having short hair, which is recessive, and brown eyes, which is dominant over any other eye color. If the breeder breeds a female with short hair and blue eyes, and a heterozygous male for both characters, long hair and brown eyes, what is the probability that one of the puppies will turn out to be a purebred with the requested characteristics? So, what do we have? So we have a female this is going to be a nightmare. I'm just going to copy and paste it. Sometimes I use my brain. So we have a female who has short hair and blue eyes, who mates or crosses with a male that is heterozygous at the loci for hair and brown eyes. So if they're heterozygous, what kind of hair color, or excuse me, hair length are they going to have? Very easy. Which unit is this, by the way? Or chapter? Oops. Long hair, exactly. Because as the question states, short hair is recessive. And if they're heterozygous, then they will have long hair. So if they're short hair, if this female is short hair, then that has to be their genotype. They have to be homozygous recessive because short hair is recessive to long, and blue eyes. And we know that because brown eyes are dominant, this is also going to be heterozygous recessive. And we know that the male is heterozygous at both loci. Okay, so Punnett squares. Long hair, what is the probability? And what we want to see from the offspring is purebred with the requested, wait, what's the requested characteristics? Oh, there it is. Having short hair, which we know has to be homozygous recessive with brown eyes, which can be any variation that has at least one dominant B allele. So this is what we want from our offspring. And what we are going to do is cross HHBB with HHBB. Wait, why do we want brown eye BB? What do you mean? Oh, it only states that oh, yes, you're right. I'm so sorry. So firstly, let's just talk about that. Yes, Carla, exactly. So we want a dog, an offspring that has short hair and brown eyes. But the question is asking for the probability that one of the puppies will turn out to be purebred while having short hair and brown eyes. And pure breeding refers to a genotype that is homozygous, whether or not it's homozygous recessive or homozygous dominant. Either of these two are technically pure breeding. So it is correct that if we wanted an offspring and we wanted it to have brown hair, it would, any B underscore genotype would be proper. However, this BB would not be pure breeding. Only this BB would be pure breeding. So this is what we want from our offspring. And we are going to cross these two individuals, the female parent and the male parent. And I think I even have this somewhere because I don't want to have to retype it. Which week would this have been? It would have been after the first exam, actually. I'm just going to not be a baby and type it out. Excuse me. So we have this H. Actually, I'm not going to explain it. I'm just going to speed run it. It's been such a long time. Oh my God. So the first possibility is HH. Maybe I can't speed run this. I'm not smart enough. H with, I'm just going to do a Punnett square. Bum, bum, bum. So we're going to have 1, 2, 3, 4 with 1, 2, 3, 4, I think. God, I don't even know how to do this anymore. Oh God. So we're going to have this HH crossing with this HB, and that's going to produce a few different H genotypes. Okay, I remember now. So I'm going to need a different table. remember, kids, you got to practice your genetics or you're just going to forget everything. So what we do is like formulate the different gametes that can form from this, and then we cross them. So HH with, wait, am I even thinking of this right? Wait, oh God, maybe I should look for it. Okay, here we go. Person contacts a pure red dog, short hair, brown eyes. Make sure it's the right question. Breeder, female. Heterozygous. Okay, so that's not the right question. Dang it. Sorry, I should have prepared this beforehand. Okay, I'm just gonna have to do it. God damn it. Shouldn't, it should be in the 16 square, I think, if it's regular dominance. I'm actually going to admit defeat and ask if Sam could please rescue me.
Sure. Oh, so basically we have to find out what our gametes are from our parents. And so from our first parent, we had gametes of HB. And because it's all recessive, they're all going to be HB. And so we don't need these 3 columns, or like you can put them there, but I don't care about them. So next we have to find the gametes of our heterozygous parent. And so when you do that, you should get HB, 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 and HB. And then you just carry out your cross and you should get, what do you get? You'll get HHB, oops, BB, HHBB. Yep. Yeah, so the whole top row, they're all going to be HBs. And so they're all going to produce the same thing down here. So everything will be the same for these 3 columns, for these last 3 columns as the first column. And I just realized you can't see me highlighting, but that's the gist of it. And so then you just have to find out what the probability of getting a, what do we want? A purebred with short hair and brown eyes. So what phenotype is that? Okay, so because this is little h, big B is the genome. Yeah, so the answer is 0. Does that make sense to everyone?
Thank you for rescuing me, Sam. Yeah.
Oh yeah, no problem.
So I guess I was right that you have to like look at an individual parent and see what kinds of gametes that they can produce, which will then go into a cross between the two. And I was panicking and not confident in myself, but we can see that this female can only give a little h and only give a little b, because those are the only types of alleles that they have. And so all of their possible gametes that they can contribute is little h, little b, little h, little b. And so then we can just look at the male and see that little h can go with big B or with little b. So little h or big H with big B, big H with little b, and then the same with the little h and the big B or little b. And then we cross those. Sorry for fumbling that. And then, as Sam said, what we want is this little h, big B genotype, because that is the only purebred genotype that can give us the characteristics that we want. And obviously, we see that 0 times here, and therefore the probability is 0. Thanks, Sam. And then the next question is the converse, which is actually this question. So this gave everyone a lot of trouble too. So what we have is a person, it's the same situation, except the person wants the same thing, the same kind of offspring. However, they are now crossing different parents. So our female was previously homozygous recessive, and now both parents are heterozygous. And instead of having to make this table and do the crosses ourselves, we know that a cross between two entirely heterozygous individuals is a special kind of relationship. It's known as the test cross, and it produces our very memorable 9:3 to 3:1 ratio of phenotypes among our offspring. So the way that we determine that is, so we see that a breeder breeds a female that is heterozygous at both loci with a male that's heterozygous at both loci. So following our last example, we have HB, heterozygous at both loci, the hair locus and the eye color locus. And what we want from our offspring is the same thing, HHBB, purebred. However, we know that this cross will produce a 9:3 to 3:1 phenotypic ratio. And so we can just look at that, and we know that in this ratio, 9 out of 16 are having a dominant phenotype at both loci. 3 of them are having a dominant phenotype at the first locus and a recessive at the second. The next 3 are having recessive at the first locus, dominant at the second. And that's exactly what we want from our offspring, because we know it has to be HH lowercase, BB uppercase, according to the desire stated in the problem. And so we know that 3 divided by 16 is what we need. And so this 3 divided by 16 gives us having recessive at the first locus and dominant at the second locus. So being recessive at the first locus and dominant at the second locus can result from having this genotype or this genotype, because this is recessive at the first locus and dominant at the second. This is also recessive at the first locus and dominant at the second. However, this individual is not purebred. And because the question demands it to be purebred, we only look at this one. Cool. Nice. Okay. Sick. Are you still eating, Sam? Do you want to do this one, or do you want me to do it? I'll take that silence as eating. Okay. So if a female AABB mates with a male AABB, and I realize that saying that out loud does not help, what is the probability of having 2 kids, of which one is AABB and the other is AABB? Let's investigate. So we have a female parent, AABB, BB, and they mate with a male, AABB. What is the probability of having 2 kids in which one is AABB and the other is AABB? So let's just do the cross. It's identical to this question that we just did, except there's not a demand for pure breeding, so it's a little bit easier. So if we do this, this parent, this female, can only give, let's also articulate that just so it's easier to refer back to. So this individual can only contribute a dominant A allele, but they can also contribute a uppercase or a lowercase. Oh, Carla, do you want to? Well, thank you for coming. If you want to put your question in the chat really fast, I can answer it and it'll be in the recording. Oh, yes, that's right. It was about frequencies. I will make sure to put that. Yes, we'll make sure to do that. Thank you. But thank you for coming, and I hope you have a great day and not a terrible time at work. But yes, so no problem. Yeah, we'll get to it. Thank you. So this female can produce this gamete or this gamete. This A multiplied by this B, this A multiplied by this B, this A multiplied by this B, and this A multiplied by this B can only give two possible gametic phenotypes. And that is crossed with this individual who is the same but in reverse. They can only give a capital B, but they can give either a lowercase or uppercase a. So they can give this gamete or this gamete. And so when we cross those together, this is 1 to 1 to 1 to 1, by the way. It's all even. And so when we cross them together, we can get, actually making a table would kind of be easier. Probably, so we're gonna need 3 rows on top, 3 rows on the bottom, or no, like this, there we go. So I was wrong, no, I was right. So this is the female on top, and this is the male on the bottom. And then we cross them. So these are all of the offspring that can result from the cross between these two individuals. We did this, or we found this out by articulating what gametes could possibly come from these parents in the first place. And then we crossed the gametes to get these resulting genotypes. And so what we're looking for is, what is the probability of having 2 kids in which one is AABB, so homozygous dominant at the first locus and heterozygous at the second locus, and the other one is AA, or homozygous at the first locus and homozygous recessive at the second locus. So what we do is find the individual probabilities of this event happening and then this event happening. So probability of having 2 kids in which one is AABB. So what's the probability that just one kid has this genotype? Based on the table, it's 1 out of 4. So we have 1 divided by 4 is the probability of having 1 AABB. And the probability of this happening twice is the probability multiplied by itself. For example, if you have a coin and you flip it, the likelihood that it's heads is 1 divided by 2. Then the likelihood that you get heads twice in a row is 1/2, the first probability, times, I've explained it before in the past, and it should be pretty intuitive. So we have 1/4 multiplied by 1/4. I'm sorry, I'm going kind of fast. We have a lot of questions. So 1/4, and that is also prob of AABB. So we found the probability of this event happening, which itself was made up of 2 events. And because it asks the probability of this event happening and this event happening, and events are multiplications of probability. And if you don't remember that, please refer to previous reviews. So now we have to multiply the probability of this event happening by the probability of the first event happening. And so we need heterozygous, homozygous recessive, and that is not present in any of our possible genotypes that result. So 0 is the probability of this event happening. And so when we multiply all these numbers together, obviously we're multiplying by a 0, which means the total probability of both of these happening is 0. Well, thank you for coming. Of course, of course. I hope you have a great day too. Lovely and fantastic and amazing. Okay, next question, if no one has any concerns about that one. Assume that a cell 2n equals 6. How many chromosomes and DNA molecules will the cell have at the end of meiosis 2? So firstly, before I answer this, what is this even talking about? What topic does this pertain to? And Sim, feel free to take over if you ever want to. Number of chromosomes. Well, yes, that is what it's asking for. But like, how do we go about doing this? What concept are we going to have to revisit in order to answer this question? Yep, very good, Casey. It is meiosis. Did you want to do this one or the next one?
I can do this one if you want.
Oh yeah, no problem.
All right, so basically we know that our cell is 2n equals 6. So what does the 2n tell us? What ploidy does it tell us about? Yep, so diploid. And so since we're talking about meiosis, what happens to the ploidy of our organism when it undergoes meiosis? Yes, the N gametes have half the number of chromosomes in DNA as our parents. And so if we're looking at if we want to know how many chromosomes and how many DNA molecules we have at the end of meiosis 2, so once all of meiosis is over, that just means that what we started with just gets halved. So we started with 6 chromosomes, which meant that we also had six DNA molecules. And we underwent meiosis, so we just have everything. And so we'll get 3 chromosomes and 3 DNA molecules. And that is that. Does that make sense to everyone? Okay, cool. Yes, so 6 represents the number of chromosomes. Yes, 3 homologous pairs. All right. Let me see. Okay, so the next one, let me see if I can copy and paste this into the doc. Okay, cool. So we are told that we have a haploid organism who has 5 chromosomes in G1 of interphase. How many DNA molecules and how many chromosomes would it have during anaphase? So after interphase comes S phase, and so that's the synthesis phase where we duplicate our chromosomes, and in duplicating the number of chromosomes that we have, we are also duplicating the number of DNA. So we have double the chromosomes and double the DNA. And so, let me take that back. We have chromosomes who have two chromatids. So they have sister chromatids. So you still have 5 chromosomes at the end of interphase, but you'll just have 2 sister chromatids for each chromosome. But then you have to know that at the end of anaphase or during anaphase, your sister chromatids are being separated. And so in separating your 2 sister chromatids, you are separating them into chromosomes and therefore doubling the number of chromosomes. I don't know if that even sounded right. I can explain that again if you guys need me to. Yeah, it's much easier to visualize what is going on. Yes, Casey. So S phase is where the DNA gets replicated. So the number of DNA molecules doubles, but you still have the same number of chromosomes because the 2 DNA molecules are part of your sister chromatids. All right, so the next one that we were asked to go over is all right, Garrett, I'm going to start sharing my screen because the next question goes off the page. All right, so we are told that a haploid organism has n equals 10, so it has 10 chromosomes. How many DNA molecules and how many chromosomes will the second polar body have? And the correct answer would be that a haploid organism can't undergo meiosis. And the reason why haploid organisms can't undergo meiosis is because if you try to split its chromosomes up, it would be missing a lot of genetic material. So let's say like you're looking at its chromosomes and it has chromosomes 1, 2, 3, 4, 5, and 6. And because it's haploid, it doesn't have any homologous pairs that go along with it. And so if you try to have that organism in or through meiosis, one of its daughter cells would have chromosomes 1, 2, and 3, but then the other one would have 4, 5, and 6. And so both of them are missing genetic information. And so like Usman said, it's already in half, so you can't split it again. So that is that one. And then the next one, I will copy and paste this into our document also. So a gene has 5 alleles. How many different genotypes are possible at this locus in a diploid organism? Garrett, I think that you were better at explaining these 2 questions. Do you want to go over these 2?
Oh yeah. So a gene has 5 alleles. How many different genotypes are possible in a diploid organism? Oh God, this is that one random slide from let me try to find it. Chapter 7, question mark.
Do you want me
No.
to stop sharing my
No,
screen?
you're good.
Okay.
This is the one about the ducks. This is the
Oh,
ducks.
it's like the hierarchy of dominance, right?
Yeah. Let me find the duck slide. Um, yeah, this isn't something that I ever really learned. So question 6 in this is asking about the hierarchy of dominance. And that's when you have different alleles at different loci. There is apparently a hierarchy of dominance among the alleles. And because of this, differing distributions of dominance within a single gene, a kind of, yeah, something like that. The exact equation is that where's the exact equation? Oh, it's not even there.
If I'm being honest, I didn't know that there was an equation for this.
Yeah, there is. That's the only way you'd be able to get the answers if you knew the equation, and we spent like
Yeah.
10 seconds talking about it. Fine, I'll look for it. I don't even know which chapter it's in. Anyway,
I
we can move
also
on to
do not.
question 7. What were you
Okay.
saying, Sam?
Oh no, I don't know which chapter it's in either, but 6 and 7 are the same deal.
Oh, okay.
And then that was all from the questions that we were emailed.
Yes.
That was the last one.
I will find that slide. But we still have 30 minutes.
Mhm.
So no rush. And we'll also get to Carla's Hardy-Weinberg question. I think it would be something like Mendelian genetics, maybe.
Yeah, that sounds like it would be it.
Maybe chapter 4, like elaborations on Mendelian genetics. Yeah. Honestly, I'd never learned about this when I was in genetics. So I'm a little bit unfamiliar, I'm sorry.
No, we didn't cover it the way that we did here when I took genetics either. We just like talked about it for a second and then that was it. But I didn't know that there was a formula and math to it.
Here it is. So if you are given a question that asks, or it gives you the number of alleles that are present within a gene, and it asks you the number of genotypes that could possibly result from this combination of alleles, the answer is this. So the number of possible genotypes are 1 plus the number of different alleles that represent a particular gene locus, multiplied by that number, divided by 2, presumably 2 parents. Let's try and decipher what this means. Oh, thank you for coming, Casey. You're always a delightful fixture of these reviews. Oh yeah, and if you guys have any questions before the final, let us know and we'll try to help. So n plus 1 I'm not going to try to make sense of this, but this is the equation. Yeah, and you can find it in chapter 5 at the beginning. Sorry that we don't know more about it. So progeny are half restricted, 1/4 mallard and 1/4 dusky. So segregation applies to crosses with multiple alleles. 3 alleles determine the type of plumage. Yeah. And you could also work this equation backward if it gives you the number of, actually, maybe not, I don't know. I mean, algebraically, you can work it backwards, but I'm not sure if that concept would still apply. Yeah, I guess that's the long and short of it. Yeah, I forgot. Let's do that guy. Some Hardy-Weinberg stuff. So calculating genotype frequencies. So when we're looking at, this is Carla's question, by the way, when we're looking at a population of individuals, obviously they all have a genotype. You know, if they're a living organism, they probably have genes and therefore they have a genotype. And so what we can do is look at this population, and if we find all of the different kinds of genotypes, we can like calculate the frequency within this population at which the genotype exists. So what we can do is look at or categorize all the individuals who have, for example, homozygous dominant genotype. And all of the individuals with the homozygous dominant genotype divided by all the people with the genotype is the frequency of the homozygous dominant genotype within the population. And then the same holds true for heterozygous individuals and homozygous recessive individuals. So this is how you'd find a genotype. Where'd my keyboard go? Oh, there it went. And then a little bit more tricky is calculating the frequency of alleles for these different genotypes that we know exist within our population. And so in order to do that, assuming that we're looking at genotypes that are simply made up of 2 alleles, what we do is that because we know that there are different dominances according to Mendelian genetics, that you can have homozygous recessive, homozygous dominant, heterozygous, we have to add these up in particular fashions. So the frequency of an allele within the population that we know is made up of a certain number of genotypes. The frequency of an allele is the number of times we see that allele divided by the number of alleles in that population, you know? For example, if we wanted to like look at the frequency of, no, that's a bad example. It should be pretty intuitive, I guess, maybe not. That's why people are asking about it. So calculating it, go forward, please. Let's do an example. So in a population of 500 hippopotamuses, 200 are spotted, which is homozygous dominant, 220 are striped, which is heterozygous, and the remainder are solid, which is homozygous recessive. What is the genotype frequency of the solid hippos? So how do we find genotype frequency? The frequency of a genotype is simply the number of individuals that exhibit that genotype divided by the total number of individuals. So we want to know the number of solid hippos, and so which genotype corresponds to a solid hippo? This one, and that is just the remainder of 500. So 220 plus 200 is 420, which leaves 80 individuals left. So 80 divided by 500 is 8 divided by 50, which is 4 divided by 25, which if you multiply that by 4 gives you 16%. So it should be D. Well, it does not give us the answer. It's probably D. Yeah. So what is the allele frequency of the dominant hippo allele? So let's not forget, let's see if I can I write without, oh, you can write without a pen. Okay. So we have 200 homozygous dominant hippos, and then 220 heterozygous hippos, and then 80 solid homozygous recessive hippos. So what is the allele frequency of this of the dominant hippo allele? So obviously we can make sense that capital A is the dominant allele. And so we need to find the frequency of this allele within this population of 500 individuals. And so we can do some very simple math. How many times do we see this capital A in this genotype? 2 times 200. Yep, Usman got it, beating me to the punch. So we see this capital A allele, 1, oops, that was a bad, 1, 2 times within this genotype. So we would have 2, because we see it twice. There are 2 alleles within this genotype, and the genotype is displayed by 200 individuals. So we have 2 times 200. And then we can add the number of times we see it in future genotypes. How many times do we see it in this genotype? Obviously, just the 1. And there are 220 individuals that have this single A. So we have 1 times 220. So 220, and we add them together. 1 times 220. And then we add the number of times we see the capital A within this third genotype. We see it 0 times in this genotype. So 0 times 80 is obviously going to be 0. We can add 0. And so this is the total number of times we see the allele in this population. How can we translate this into a frequency? Ah, Usman got it. So the frequency of the frequency of an allele is the number of alleles divided by the total number of alleles in the population. So the frequency that we're looking for is the number of alleles that we're looking for divided by the total number of alleles that exist in the population. And so there are 500 individuals in this population, and each individual has 2 alleles as a part of their genotype. So there are a total of 1,000 alleles in the population. And so this is 400, 620 divided by 1,000 is 62%. That was a very simple example. I remember more complicated ones on Dr. Chiari's exams, but that should suffice for now. Just try to keep your stuff straight. Do we have any other questions? Let's see. Nick, do you happen to have a question? Usman. It's just the 4 of us. Oh, well, thank you for coming. You're awesome.
Thank you so much for being here.
Usman is always here. Usman's a champion.
Yes.
And you're always here too, Sam. You're a champion.
Ah, thank you. You're also champion because there's always
What?
2 of you here.
Okay, well, if we don't have any more questions,
You all are free to go.
Yeah, thanks for hanging out this semester.
My goodness, good luck to you two guys. Keep in touch. We would love to hear from you later on through email, or like if you see us on campus, say hello.
Yeah, and I think I might have some classes with you guys next semester, like organic and anatomy.
Gosh, don't sound too excited. Oh my goodness, yes. Thank goodness we have Dr. Veretto now.